I worked for the last 40 years at the Rowett Research Institute in Aberdeen. I no longer work there, but this is one of the premier nutrition research institutes of the world. Well, certainly is one of the largest nutrition research institutes, but it is mainly animal nutrition. There is a human component in it, but it is mainly animal nutrition. Now that, in a sense, predetermined the sort of things which we are doing. I was always interested in plants and crop plants and legume plants. We were very interested in particular parts of the plant constituents which are normally called anti-nutrients. These are compounds which are present in plants which don't do much good to us. They have a nutritive value, but because of some structural or other features, enzymic features, they are not doing us much good. But because these are still food and we eat them, it came to the attention of the biotechnologists soon that what is possibly a problem for us may certainly be a problem for the insects. And this is our entry into genetic modification, quite particularly taking some genes, lectin genes, either from plants, higher plants or lower plants, bacteria, and use them in that context to see what is the effect of these potentially harmful effect on insects. When the genetic modification business came along, then we had this sort of study extended to insects. Quite literally, if it does do some harm to the insects, alimentary tract or digestive system or whatever, and if it is not doing similar harmful things to the higher consumers like mammalians, then we'll be in business. So our job was, in a sense, to identify potentially useful natural insecticidal genes in plants, those which would not harm us as higher consumers of the same crops. We had a very big research group, usually between 18 to 20 people, because the European Union sent, we were an accepted teaching laboratory, and they sent a lot of postdocs to us with all the money that they all had. And this research group was headed since 1990 by my wife and colleague, Dr. Susan Bardot, and we sort of agreed at the beginning, not just with her, but with the other members of the team, that this would be a good adventure for us. We really thought that this was a great idea. We're going to look at it. We'll come up with, based on our experience with non-GM foods, we will come up with a credible and scientifically solid risk assessment procedure. In fact, that nearly $3 million research grant, which we did get, which was for 3 institutes collaborating in this project, and I coordinated the project. This was to draw up a blueprint for risk assessment, which we work it out on a a model, which in our case was the genetically modified potatoes, but it would have general applicability. So the regulatory authorities can use this method and demand that sort of data from the companies in order to evaluate the safety or otherwise of that particular genetically modified crop. If we, for example, find a gene whose product is likely to be very detrimental to a target insect, which we regarded as a pest, and which it would not damage us, or the damage would be minimal, then the establishment of this would serve as a good blueprint for risk assessment. So this is why we really started. In our case, it was with the potatoes. We had in Scotland, there is a major problem with aphids. And the aphids are controlled under normal conditions, what we call ladybirds, and in your case you call them ladybugs. So we extended the studies even to this, what we call scientifically tritrophic interactions, where we looked at the, okay, we did the genetic modifications, the plants were growing in the greenhouse, we release aphids, and so we can measure the actual damage, and then we release ladybugs into the same system and see how they are affecting the whole system. Would the ladybugs be affected? You know, this is, it was the days before Losey's monarch butterflies, and there were quite a number of studies, like in Switzerland, to see if apart from the target insect, the target pest, shall we be doing any damage to other things which we consider the good guys. About a year and a half into the project, the storm clouds started to gather. One of the first ones was not by us, but by the entomologists. You see, that in science, you normally have, if you put in a gene and a gene product is expressed, the effects of that should correlate with the amount of expression. In plain language, if you have a method of estimating, let's say, on a leaf, what is the expression of that gene product, and then you put it into this system of releasing the aphids, and you measure the damage or the protection which the gene conferred on the plant. If you correlate protection with the expression level, you must have a good correlation. You plot it, you will have to see if there is more expression, you must get more protection. Now that was the first hole in the whole system, that it didn't work out that way. What happened was that some plants which had very little expression were better protected against the aphids than some of the other plants which had high expression levels. Now this was the first time when we started to think about it, that is this one gene, one protein, there is something wrong with it. So the way how it is inserted into the plant genome is probably far more important than the expression level. And then things started to come from there on. Then we found these people, entomologists, they found that all right, the aphids were, in a sense, poisoned by the expressed gene. But when we released the ladybugs, they were also poisoned because the ladybugs were eating the aphids, and the gene product got into their digestive tract. So now that's not much good, is it? I mean, you're just simply upsetting nature, the natural balance. So what we did was that we first we took the genetically modified potatoes and put as much as possible of this into the diet, and we fed rats on it for a short time, 10 days. That's an appropriate time in most of the nutritional studies as a sort of preliminary short-term study. And we found that there were some problems. And then we said, oh, but is it possible that if we dilute it with a good protein, a non-GM protein, would these problems disappear? Would you dilute them out? So when we did that, we found that no, we didn't. The problems persisted, and particularly the problems affecting the gastrointestinal tract of the rats. The problems were that the genetically modified potatoes induced what we call a proliferative growth in the small intestine, and I shall explain what it means. Now, but before I do that, the most important thing was that we pre-selected the gene that its product should not do that. So we spent 6.5 years of selecting out a gene whose product wouldn't do the thing which we did see in the genetically modified potatoes. And you may get, like with the flavor-saver tomatoes, those female rats develop pinpoint erosions, sort of pinpoint bleedings into the stomach. Now, that is, it may be temporary, and if you withdraw the agent, in this case the flavor-saver tomato, it may disappear. But we don't know, and it has to be investigated. The secondary effects can, for example, affect the liver. And we found some secondary changes in the liver, just like as this recently published Malatesta paper, which did establish that by feeding young rats on Roundup-ready soya, you do get some proliferative, again, sort of higher metabolic ranges of effects in the liver of these animals. Again, we don't know is it pathological? Would it be reversible? Would it? But you have to start somewhere. You cannot start at the end. You have to start at the beginning. Remember that all these studies are done before you can do any experiments with humans. Now, in this case, of course, the whole thing has been changed around because you're being used as the guinea pig. They are doing the experiments on you, and what is even more, you don't know whether they are doing it or not, because in the US, for example, it's not labeled. So therefore, you don't know whether you're eating GM food or not.